Hello, everyone. Thank you, Maricelis, for the invitation to give this lecture. My name is Mariam Aoun, and I am an assistant professor in small grains pathology in the Department of Entomology and Plant Pathology at Oklahoma State University. For today's lecture, I'm going to share with you some insights on the application of genome-wide association mapping in wheat improvement. This is the outline of our lecture. I will start by giving you an overview on the association mapping, then on different methods to reduce spurious association in the GWAS, some examples related to the application of GWAS in wheat breeding, and suggested method to increase the power of detection in the GWAS, and finally take-home messages that are in a form of a quiz. Starting with the first part, association mapping is also called genome-wide association mapping or genome-wide association studies or linkage disequilibrium mapping. It is based on revealing correlations between phenotype and genotype in a natural population. The main two components in a GWAS are the genotypic data and the phenotypic data. The genotypic data consists of markers that are distributed across the entire genome. The most commonly used type of markers are single nucleotide polymorphism markers, or SNPs, and these markers are used as independent variable in the GWAS model. The second component is phenotypic data, and here I am giving you example of traits. And the most important thing is to have phenotypic variation in your population for your trait of interest to be able to conduct any statistical analysis, including GWAS. And the phenotypic data is used as a response variable in the GWAS model. These are the different steps to conduct the GWAS. The first is to select diverse germplasm collection. The population size, it varies between studies, but the higher the population size, the better. This is because small population size results in lower detection power of loci that are associated with your trait of interest. And also, we may have inflated effects of markers in a small population. Then you will have your phenotypic data combined with your genotypic data and use some software to identify marker trait associations that are of interest to your trait. These are examples of phenotypic data and traits that were used to perform GWAS, plant height, disease resistance, in this case, so the stem rust, the spike architecture, seed size. So the first thing that we need to think about when doing your phenotypic data is to have an experimental design. Then collect high quality phenotypic data. This can be used using conventional methods, also high throughput phenotyping methods. And visualize your data to be able to check for outliers that sometimes can be caused by errors. So I highly recommend you visualizing your data using different graphics like histograms or box plots. And this will allow you to check if you have any problems in your phenotypic data. Then perform some basic statistical analysis like mean, variance, standard deviation, etc. And then calculate the heritability of your traits. For the genotypic data, as I mentioned earlier, we need markers that are distributed across the genome, and we need high density marker. Always higher number of markers are preferred. These are the different genotyping platforms, the high throughput genotyping platforms that are available. As I mentioned, SNP markers are the markers that we are using nowadays. Technologies like genotyping by sequencing or Illumina with array, whether it's 9K or 90K. We have also some Axiom with array. And all of these genotyping platforms should provide high quality genotypic data and good coverage of the genome. The marker data should be filtered by removing monomorphic markers. Also, markers with high missing data and high heterozygosity levels should be removed from the analysis. We need to exclude markers with low minor allele frequency because those can cause false positives later in the GWAS. And for some software, missing data is not allowed, so that's why imputation of missing data can be done using different software. The GWAS is based on linkage disequilibrium. It is defined as the non-random association of alleles at different loci within a population. So the most commonly used way to estimate the linkage disequilibrium between markers is based on R-squared, which is the squared correlation between markers, and that ranges between 0 to 1, with markers that are physically close to each other having higher linkage disequilibrium. You can estimate linkage disequilibrium across the genome, as shown in this figure. Also, you can do this in each subgenome, the subgenome A, B, and D. And also, you can do it within each chromosome or between pairs of markers. So this figure shows an LD decay across the genome where you can see reduction, or the LD is reduced as the genetic distance or the physical distance between markers increased. The threshold for the LD can be calculated using different methods that you can find in the literature. And that will help you to determine a genetic or physical distance that will help you to define whether the markers that are identified within a chromosome correspond to a single locus or multiple loci, as shown in this Manhattan plot. Here on chromosome 4A, we have 4 markers that were in high LD. So we have R-squared that is close to 1 between these 4 markers. So this shows that these 4 markers are likely associated with a single causal gene or locus. In this slide, I am showing you the advantages and disadvantages of association mapping in comparison to linkage mapping in biparental populations. So for association mapping, we don't need to develop a population. So we can use existing natural population, while in linkage mapping in biparental populations, we need to develop a cross between 2 parents and then develop recombinant inbred lines or double haploid, and that's the time consuming. The association mapping can detect many loci that are associated with the trait of interest in the natural population, while only segregating loci between the parental lines that are associated with the trait of interest can be detected in a biparental population. The GWAS, or the association mapping, have higher mapping resolution, and that's because we have higher recombination events in the natural population compared to fewer recombination events between the 2 parents in a biparental population, which results in limited mapping resolution in linkage mapping. Because we have high number of or recombination events in a natural population, that's why we need higher density of markers to detect these recombination events. While for linkage mapping, because we have larger blocks of LD, then we don't need as much markers as it is in the association mapping. So it is not helpful to have high density markers in biparental population as it is in the GWAS. One limitation of the GWAS is the detection of rare alleles in natural population in comparison to higher detection power in biparental populations. And I will discuss with you later in this lecture how we can have other methods to increase the power of detection of rare variants. Coming now on how we can reduce spurious association in the GWAS. Population structure can lead to false positive in the GWAS. This is an example of a Genome-wide population coming from different breeding programs. And you can see based on principal component analysis that you have grouping of like 5 groups in this population. And also the same grouping was observed in this dendrogram. So population structure can be due to other factors that are not related to your trait of interest, like adaptation or diversifying selection in different breeding programs. So that's why it's important to include population structure as a Q matrix in your GWAS model. And that will help reduce false positives in the GWAS. The most commonly approach that is used to estimate the Q matrix is based on principal components. So how many principal components you include in your Q matrix? We usually need to use the first few principal components that explain most of the variation. Here in this example, we can stop at PC number 3 because we have plateauing in the variance that is due to the other principal components. So scree plot can help you visualize how many principal components you need to include in the Q matrix. Family relatedness also is another cause of false positives in the GWAS, which is due to co-ancestry between individuals in your population. So this heat map is showing you how these individuals in this population are related by calculating this identity by state matrix, which is based on marker data. If you have pedigree information, then you can calculate an identity by descent matrix, which is basically how these individuals are correlated. So here, where you see some red spots in the off diagonal, those are individuals that are highly correlated with each other. And I usually exclude the individuals in my population that have high correlation, or where you can see an identical SNP data in like these lines are called differently, but they have exactly the same SNP data, and those duplicates should be excluded from the analysis because they tend to inflate the effect of some markers. Here I am showing you the equation for mixed model. So where the phenotype is used as a response variable, you have the component for your SNP markers, the population structure, the Q matrix that is included as fixed effect, and the kinship or the K matrix that is included as random effect. And this is for the family relatedness and the error term. There are many software that can be used to perform the GWAS, like GAPIT, Tassel, Genomics, GEMA, and P-Link. I personally prefer using free available software like Gapit and Tassel. There are multiple GWAS models that you can test for your trait, and then select the most appropriate model to report your results. Single locus GWAS model, the most commonly used is mixed linear model. However, a limitation with mixed linear model is the power of detection of marker trait associations. So that's why multi-locus models are suggested. An example of that is farm CPU, where this type of models have higher power of detection while also controlling false positives. How can we select the most appropriate model? We have different criteria. The first is the quantile plot, which is shown in here, where you plot the observed p-value versus the expected p-value. And the best model will be where this distribution is close to the diagonal, which means that we have good control of false positives or false negatives in the model. Here are QQ plot for 4 models, naive model, where none of the population structure or family relatedness were included. GLM, where only population structure was included. Compressed mixed linear model and farm CPU models were the most appropriate in comparison to GLM and naive model, where we have a higher rate of false positives. You can also use other criteria like mean square difference, the BIC and the AIC. So the lower this statistics, the better is the model. In this table, I am showing you example of different GWAS model that you can test, both single locus models like GLM, mixed linear model, and compressed mixed linear model, and multi-locus models like mixed linear, multi-loci mixed model, super farm CPU, and D-Link. Here you can see whether they account for population structure and kinship, how they do in terms of controlling false positive and false negatives, and the computational time, which it is important, especially if you are running several traits. And these are how commonly these models are used. Mixed linear model is an old model, so that's why it is the most commonly used model. And farm CPU is also a commonly used model. I encourage you to go to this tutorial in Gapit. You will have more details related to these models, and also tutorial to show you how you can run each of these models in Gapit. In the output of your GWAS, you will have p-values that are associated with each of your marker that you included in the analysis. And to control for false positives, there are multiple testing corrections that were suggested, like the Benforoni correction and the false discovery rate. And these are the most commonly used ones, but there are other corrections that can be used. I personally prefer using false discovery rate because it is less stringent compared to the Benforoni correction. So the adjusted value for the Benforoni correction will be the p-value for each of your marker divided by the total number of markers that are used in the GWAS. And this is considered a stringent cutoff and can lead to false negatives, where you can lose some true associations. So here is an example of an FDR correction cutoff of 0.05, that was done in the GWAS for stripe rust resistance in wheat. Coming now to some examples of the application of GWAS in wheat breeding. Here I am showing you publications of for GWAS in wheat from 2009 until 2020. So you can see this very rapid increase in deploying GWAS in wheat. And here are the number of these studies based on these trait categories: biotic stresses, abiotic stresses, agronomic and yield traits, and quality traits. But most of the studies, they were done for biotic stresses and agronomic and yield-related traits. For biotic stresses, half of these studies, they were done for looking for loci that are associated with rust resistance in wheat. GWAS are used mainly to identify genomic regions that are associated with various traits so that we can use these markers in marker-assisted selection. Remember that GWAS can provide markers that are tightly linked to the causal gene. So that's why they can be useful in marker-assisted selection in comparison to markers from biparental populations where the mapping resolution is lower. So here are two examples of GWAS that were done for leaf rust resistance and for a quality trait, which is the single kernel hardness. Here I am providing you the papers, and you can go and read in more details on how GWAS was used in these studies if you are interested in applying GWAS in your research. Here are two examples on the application of GWAS for yield-related traits, tiller number, and the spike length. So once you identify significant markers that are associated with your trait of interest, and especially if these markers have large effect, then they can be converted into user-friendly PCR markers like competitive allele PCR markers, the CASP markers, or START-P marker, the semi-thermal asymmetric reverse PCR markers, where you can distinguish between the two homozygotes group and the heterozygote group by simply running PCR. Another way to use GWAS is to identify candidate genes. This is an example on how GWAS was used to identify candidate genes that are associated with spike-related traits. So here is a QTL on chromosome 4A, I believe, that was associated with the spike length and the grain number and with the grain setting. And in this locus, they looked for the annotated genes that are within these genomic regions of these associated markers, and they found 11 genes within this genomic region from the International Wheat Genome Sequencing Consortium website. And this could be done thanks to the available genome sequence that is there and the annotated genes that you can find in this website. Then they were able to refine the candidate genes into 2 genes, and that's by looking at the gene expression data in this website, and also where you can look at the gene expression of these genes in different tissue and also at different developmental stages. Another example on how GWAS was used to identify candidate genes it was was described in this study, where this gene TAHXK3 on chromosome 2A was identified as a candidate gene controlling stomatal index in wheat, which is associated with response to drought. So here again is a equatorial that was identified on chromosome 2A, and there is zoom on this locus and the annotated genes that are available on IWGSC website. And again here, by using some gene expression data, they were able to determine this gene as a candidate gene that is associated with stomatal index in wheat. Gwas was also used to assist genomic selection. I believe that in this course you had a lecture related to the application of genomic selection in wheat. So briefly, you have a training population that is phenotyped and genotyped, and that will be used to develop your genomic selection model. And then your breeding population that was only genotyped. And based on the genotypic data, and the genomic selection model that you develop in your training population, you will be able to predict the genomic estimated breeding values of the individuals in your breeding population, and be able to select the best individuals in this population. How GWAS can help in this case is the significant markers from your GWAS, they can be fitted as fixed effect in your genomic selection model. And this shows to be effective in increasing the accuracy of prediction. Here is an example on how Gwas-assisted genomic selection helped to improve the prediction accuracy for resistance to Septoria TTC blotch. In this table, you will have the number of fixed effect markers from 1 to 5, and these markers were fitted whether because they were significant in the GWAS, and to compare the effect, they also selected markers that were randomly selected. So in the randomly selected markers, you don't see any improvement in your prediction accuracy. This is R equals 0.44. While in the case where you included markers that are significantly associated with the GWAS as fixed effect, you can see this increase in the prediction accuracies as you increase the number of markers. So the accuracy went from 0.47, where no markers were included as fixed effect from the GWAS, to 0.62 when 5 markers that were associated with Septoria TTC blotch were included. Going now to the methods that were used to increase the power of GWAS. So instead of using natural populations, and I talked earlier in this class about the limitation of GWAS and natural population is the limitation to detect rare variants. So doing GWAS in a nested association mapping panel or in a multi-parent advanced generation intercross MAGIC population, this can improve the power of detecting rare alleles in GWAS. So for the nested association mapping, you have one common parents. Here I'm showing an example of 4 biparental populations that were crossed to different parents. And then you will do selfing to develop recombinant inbred lines. And then the GWAS can be performed in this recombinant inbred lines that are developed from these biparental crosses. Also, you can do linkage mapping in each of the biparental cross. For MAGIC population, you have different parents that will be crossed. In the next generation, you will cross the F1 and so on until you do selfing, and then you develop recombinant inbred lines. Another method that was proposed is to use haplotype-based GWAS instead of the SNP-based GWAS. So one limitation of the GWAS is that it doesn't detect the epistasis that is present between markers, the interactions between SNP markers. And this will lead to a small proportion of the genetic variants that will be explained by the significant markers. So that's why haplotype-based GWAS that accounts for epistasis between the marker was proposed to improve the power of detection. And in this figure, I am showing you example of 6 individuals where you can see here each column, the highlighted column corresponds to a SNP. And then the haplotype will be a group of these 3 SNPs. So in this individual, we can detect 3 haplotypes: the red, the AGT, the GTA, and the orange haplotype, the A, G, and A haplotype. So here I'm showing you an example of how haplotype-based GWAS can improve the power of detection compared to the SNP-based GWAS. And this study that was done in leaf-rust resistance, where the SNP-based GWAS allowed to identify fewer associations compared to the haplotype-based GWAS. And these are examples of 3 known leaf-rust resistant genes that were detected. LR22A and LR34, they were detected using both methods, while LR10 and LR1 were detected only in the haplotype-based GWAS, while they were not there in the SNP-based GWAS. So with that, I come to the end of this lecture, and these are the take-home messages that I would like you to get from this class. The first is, what is a GWAS and linkage disequilibrium? What are the advantages and the disadvantages of the GWAS in comparison to biparental populations? What are the different GWAS models that we can use, both single locus models and multi-locus model? Ways to increase or to select the best GWAS model, and how can we reduce false positives in the GWAS? Some examples on the application of GWAS in wheat improvement, and what are the methods that were described in this class to increase the detection power of the GWAS. Thank you for listening.